Here is a 3D muscle diagram. Shown in the orange is a nerve, and then we are going to go through what the muscle is made up of. The muscle is made up of fascicles. In those fascicles are myofibers. Inside the myofibers are myofibrils. Then shown are the myofilaments. There are 3 thin filaments called actin, troponin, and tropomyosin. The thick filament is called myosin.
In this diagram, the white dots represent the calcium. The green is the sodium, red is acetylcholine, the blue is the acetylcholinesterase, and the purple is the calsequestrin. Now that we've discussed the parts of the muscle, now let's go over muscle contraction. Action potential reaches the synaptic bulb, and this is the electrical reaction. Calcium channels open and release the calcium, which stimulates the synaptic vesicles of the acetylcholine to fuse with the cell and exocytosis into the synaptic cleft. The acetylcholine is picked up by the receptors in the motor end plate, which opens up the channels for sodium to flow into the skeletal muscle. The action potential travels down the sarcolemma into the T-tubule, which stimulates calcium release from the terminal cisternae, where the calsequestrin keeps hold of the calcium. Calcium then bonds to the troponin, which causes the tropomyosin to uncover the active sites of the actin. When the active sites are uncovered, ADP is released, and high-energized myosin heads bond to the active sites, forming a cross bridge. This power stroke causes the thin filaments to slide over the thick filaments. ATP is needed for the myosin heads to break free from the active sites. Now that we've talked about the neuromuscular junction, let's talk about how the muscles contract. The sarcomere is the unit that allows contraction. It extends from Z disc to Z disc and contains thick filaments, which are the myosin, and the thin filaments, that are the actin. The A band extends from the length of the thick filaments. The I band is where there are no thick filaments. The Z disc is where the thin filaments attach, and the M line is where the thick filaments are attached. The H band is where there is no overlapping of thin filaments. In order for a muscle to relax, the calcium will return to the terminal cisternae. This allows the tropomyosin to recover the active site. Sodium is returned to the synaptic cleft, while potassium is returned outside of the cell. Acetylcholine is broken down by acetylcholinesterase.